Your heart beats, your lungs breathe, and your brain thinks. Yet these actions are not the same kind of knowledge. One tells us what the body is made of, the other explains how it works. To understand life itself, science had to learn the difference. Welcome to Mattermosaic, piecing together the universe, one discovery at a time. Today, we are exploring anatomy versus physiology, the two sciences that together reveal the structure and function of the human body, and how understanding both transformed medicine, biology, and our understanding of what it means to be alive. For most of human history, the human body was mysterious, sacred, and largely untouchable. Early civilizations observed wounds, broken bones, and illness, but internal structure remained hidden. In ancient Egypt, around 1600 BCE, medical texts like the Edwin Smith Papyrus described organs and injuries, yet explanations focused on visible structure rather than function. The heart was seen as central, but circulation was not understood. Anatomy existed as observation without explanation. In ancient Greece, the separation between form and function began to emerge. Around the 4th century BCE, Aristotle studied animals on the island of Lesbos, comparing organs across species. He carefully described structures such as the heart, lungs, and blood vessels, laying foundations for comparative anatomy. However, Aristotle believed the heart was the seat of intelligence, showing how anatomical observation without physiological evidence could mislead understanding. Anatomy advanced through description. Physiology lagged behind because it required experimentation. The two sciences grew at different speeds. In the 2nd century CE, the Roman physician Galen worked in Pergamon and later Rome, dissecting animals such as pigs and monkeys. Galen mapped muscles, nerves, and organs with remarkable accuracy for his time. His anatomical texts dominated European and Middle Eastern medicine for over 300 years. Yet many of his physiological conclusions were wrong. He believed blood was consumed by the body rather than circulated, because he had no way to observe living systems internally. For centuries, anatomy was about form. Physiology was speculation. This changed during the Renaissance. In 1543, in Padua, Italy, Andreas Vesalius published De Humani Corporis Fabrica. Through systematic human dissection, Vesalius corrected hundreds of Galen's anatomical errors. He demonstrated that structure could be verified directly, not inherited from authority. Anatomy became visual, precise, and grounded in evidence. Yet even Vesalius could not fully explain how organs worked together in living systems. The turning point came in 1628, in London, when physician William Harvey published De Motu Cordis. Using careful measurements and observations, Harvey demonstrated that the heart functions as a pump, and that blood circulates continuously through the body. This was physiology in action. It explained function through experiment, not appearance. Anatomy tells us the heart has chambers and valves. Physiology explains how pressure gradients and rhythmic contraction move blood through closed loops. One without the other is incomplete. As microscopy advanced in the 17th century, the relationship between structure and function became clearer. In 1665, Robert Hooke used a microscope in London to observe cork and described tiny compartments he named cells. Shortly after, Antony van Leeuwenhoek in the Netherlands observed living cells, blood cells, and microorganisms. Anatomy now extended into the microscopic world. Physiology followed closely behind. Scientists began to ask how cells worked, not just what they looked like. In the 19th century, French physiologist Claude Bernard formalized this relationship. Working in Paris, Bernard introduced the concept of the internal environment, later called homeostasis. He showed that organs function together to maintain stable conditions inside the body, regardless of external change. This insight unified anatomy and physiology into a single systems-based understanding of life. Anatomy became the map. Physiology became the motion. The nervous system offers one of the clearest examples of their partnership. Anatomically, the brain is divided into regions such as the cerebrum, cerebellum, and brainstem. Neurons form networks, and axons connect distant parts of the body. Physiology explains how these structures generate electrical signals, transmit information, and produce perception, memory, and consciousness. In the late 19th century, Spanish neuroscientist Santiago Ramón y Cajal used staining techniques developed in Italy to reveal the structure of neurons. He showed that neurons are discrete cells, not a continuous network. This anatomical discovery allowed physiology to explain signal transmission through synapses, changing neuroscience forever. Similarly, anatomy identifies alveoli in the lungs as tiny air sacs. Physiology explains gas exchange, diffusion gradients, and how oxygen enters the bloodstream while carbon dioxide exits. Structure determines possibility. Function reveals consequence. The distinction becomes critical in medicine. A physician examining anatomy may identify a blocked coronary artery. Physiology explains how reduced blood flow leads to ischemia, tissue damage, and heart attack. Surgery restores structure. Drugs modify function. Effective treatment depends on understanding both. Modern biology continues to integrate these disciplines. In the 20th century, advances in biochemistry and molecular biology revealed the physiological processes inside anatomical structures. The discovery of DNA's double helix in 1953 by James Watson and Francis Crick, building on X-ray diffraction data produced by Rosalind Franklin in London, linked molecular structure to biological function. Genes are anatomical information stored chemically. Their expression is physiological activity. Medical imaging technologies further blurred the boundary. MRI, CT scans, and PET imaging allow scientists to see structure and function in real time. In hospitals worldwide, doctors now observe blood flow, neural activity, and metabolic processes inside living patients, a union of anatomy and physiology once unimaginable. Even at the smallest scale, the distinction remains essential. A muscle fiber has a visible structure composed of sarcomeres. Physiology explains how actin and myosin interact, using ATP to generate force. Without structure, there is no movement. Without function, structure is meaningless. Evolution reinforces this relationship. Structures evolve because they perform functions that enhance survival. Feathers first evolved for insulation in dinosaurs before enabling flight. Anatomy precedes physiology, but physiology gives anatomy purpose. Understanding the human form requires seeing it as dynamic, not static. Anatomy freezes the body in time. Physiology restores motion. Anatomy and physiology are not competing sciences. They are complementary perspectives on the same reality. One asks what the body is, the other asks how it becomes alive. Together, they reveal that life is not defined by shape alone, nor by activity without structure, but by the continuous interaction between form and function. To know the human body is to see both the map and the movement, the architecture and the action, and to understand that life exists only where the two meet. This is Mattermosaic, piecing together the universe, one discovery at a time. If this exploration sparked your curiosity, tap like, share it with someone who loves science, and let us know what you want to uncover next. Subscribe for more weekly journeys across the fabric of reality.